Right, so polysaccharides. What's a polysaccharide? It's a polymer. So polymer is more than what? More than two, more than three, more than one. It's going to be more than two. So three or more. Of what? Is it amino acid? Is it nucleotides? Is it sugars? Saccharide. Saccharide always means sugar. So polysaccharides means sugars joined together, more than two sugars joined together. Now, if you have lots of sugars joined together in plants, we call it starch. Sometimes plant, they also store lots of sugars together to help the cell wall, and that has a special name, cellulose. We humans, when we have lots of food, we have excess glucose. Now we need to store that glucose, so that glucose gets joined together and it forms a long chain called glycogen, and we store that in our liver, and we can store that in our muscles. So therefore, when we need it, they can be broken down by which reaction? Hydrolysis. For which reaction? For respiration to take place. Okay, so we'll be going over starch, glycogen, and cellulose. Have a look at starch. As you can see, it's a long molecule, polysaccharide. Starch, there are two types of molecules. Glycogen, have a look at that, as you can see, and then cellulose. Okay, so starch. Now, starch is the main storage of carbohydrate. What's carbohydrate? Sugars. Remember, sugar, carbohydrate is an all general term. So starch is the main storage of carbohydrate. How did these plants get these carbohydrates in the first place? How did they get that sugar in the first place? It's through which reaction? Right, photosynthesis. So plant use this starch and they store it in their cells. When they store it in the cells, that's a lot of space. In a plant cell, there's lots of chemical reaction, lots of water. So it's very, very important that that starch that's being stored does not disturb the chemical reaction, does not disturb the fluid movement, the water movement. That's why starch is fundamentally important, that it has to be insoluble. If it's soluble, it affects water potential. It affects osmosis from the cell, outside the cell. It affects the reaction. So it is insoluble, so it can be stored with so many starch and will not affect water potential, so will not affect osmosis, will not affect chemical reaction, or will not drain the cell and will not shrink the cell. Starch can be used, it can be broken down, by hydrolysis, because sometimes the plant need to use glucose for respiration. There are two forms, there are two types of polymers, amylose and amylopectin. We are still talking about starch. There are two types of polysaccharides which makes starch. One is amylose and one is amylopectin. Do we need to know that? Yes, we do. We need to know that really well. So both polymers, which polymers? Amylose and amylopectin, both of them are made from alpha glucose. You also need to know that alpha glucose is used to make starch. So amylopectin and amylose, both of them are made from alpha glucose. So amylopectin is a polysaccharide, and that amylopectin is a long chain. Okay, and it is branched, long branched. So if it's branched, it's got 1,6 as well, glycosidic bond. If you refer to the old video, we said the moment it branches off, that branch will be a glycosidic bond, obviously, between every sugar, but it's 1,6, carbon number 1 and carbon number 6. The amylose is also long, but the only difference is there's no 1,6, which means there is no branching. Okay, it coils up into a spring shape. So I'm showing you here a amylopectin, and you see the amylopectin there? Fine. All of these are individual sugar molecules joined together. So these molecules are joined together by 1,4 glycosidic bond. Look at over here. That's a branch there, right? You agree? That's a branch there. So that branch over here is 1,6 glycosidic bond. Amylose here, and the other hand, is a unbranched. There's no branches coming out. It's like it's coiled, but it's not branch. There's no branch there. So it's got 1,4 and it does not have 1,6 glycosidic bond. Both of these together, they make starch. And also, don't forget, all of these sugar molecule, all of these monomers, these individual glucose, are they alpha or beta? All of them. All of them are alpha. So let me quickly talk about amylose. Okay. So amylose, we said that obviously is made from alpha glucose. Now all of the links are 1,4 glycosidic bond. I'm showing you here a amylose. Obviously it's coiled, but I uncoiled it to make it into a straight chain. These are 1,4 glycosidic bond. So the amylose, it forms like a alpha helix. Okay, it coils into an alpha helix, and the alpha helix, that shape, is maintained, is held by hydrogen bond. Exam question: why is it that it is coiled together? What's the purpose behind it? The purpose behind it is, remember, we are storing so many starch in cells, so we need space, so it makes it compact and stores it into a smaller space. Right, let's now talk about amylopectin, the other one. Remember, it's got a branch, so it's got 1,6 as well. So I'm showing you here amylopectin. So amylopectin here, look, over there, it's not branched, it's straight chain, okay? So these are 1,4, 1,4, 1,4, 1,4. Then I've got a branch coming out. So that one there is going to be 1,6 glycosidic bond. So amylopectin has branch structure, okay? So the side chains coming off are 1,6 glycosidic bond. Exam question: why is it branched? The reason why it's branched, okay? So remember, this starch molecule here is stored for a reason. Plant stores starch for a reason. They don't hoard it forever and ever. Plant, they need glucose for respiration. So now they're storing that starch. Remember, individual of them, all of these are glucose molecule. So let's say the plant needs glucose. So then what happens? We have hydrolysis occurring. Water is put in, and that one glucose at the end comes off. Okay, and it's used by the mitochondria for respiration. So the reason why it's branched is that to increase the surface area, because when the enzyme comes to break that bond, that glycosidic bond, using hydrolysis reaction, the enzyme can do it more quicker if they have more areas to work with. So having a more branch, you have different space, you have increased the surface area, enzyme can hydrolyze that glycosidic bond more quickly and release that glucose more quickly, so therefore you have a faster, quicker respiration occurring. Okay, let's go back to animals. So glycogen. Glycogen is what is storage, is a polysaccharide of lots of glucose together, found in animals, in the liver, in the muscles. So glycogen is, the structure of it is a bit like amylopectin with regards to, like, it's got branches coming off. So if you've learned amylopectin, glycogen should be easy. Exactly same thing, just change the plant into an animal. So it's made from alpha glucose, and then you have the side chain is 1,6 glycosidic bond, and we store it in our liver, in our muscle cells. Animals store excess glucose as glycogen, as we mentioned. So glycogen, again, the question arises, look at glycogen here. It's really, really branched. Fine, it's got more branches than amylopectin, but still branched. So these side ones over there, it's got 1,6, and all of these ones are going to be 1,4. So question arises, why does it have to be branched? Exactly the same thing. We humans and animals as well, they require lots of respiration. Let's say you're really, really hungry and your body needs energy, so you need respiration. You have oxygen there, that's fine, but you don't have glucose, there's no food. Oh, you have glycogen stored? Where is it stored? In your muscle, in the liver. So glycogen, let's break it down. So the enzyme is going to go and break down that massive molecule of glycogen, polymer, into small bits using hydrolysis. So having lots of branches mean the enzyme can hydrolyze it faster. How is it faster? There's more access points, more surface area. So you can release that glucose more quickly, so therefore you can undergo more efficient respiration. And second point is that glycogen is branched, so therefore it's a compact molecule, so it's good for storage as well. Now, same point as starch. Glycogen, our liver and muscles, they are storing glycogen. So do you want that glycogen to affect osmosis, to affect water potential in your body? No. So do you want it to be insoluble or soluble? You want it to be insoluble. So glycogen is stored in our body, but it's insoluble. So therefore it does not affect water potential, does not affect chemical reactions. Right guys, we are almost done. Last one, cellulose is also a polysaccharide. Again, it's to do with plants, but a particular part of plant, which is the cell wall. So cell wall is, as you know, is a major structural component. Cellulose is a major component of the cell wall. If you don't have cellulose in your cell wall is not a wall. It's a weak, fragile wall. Your plant will not survive. So the cell wall has to be really, really strong. Has to have strength and stability. So it provides support and allows the cell to be turgid. And now cell wall is, as you can see here, is made out of beta glucose. So far we've been talking about alpha glucose everywhere. This is the first time, beta glucose. So the OH, is it pointing downwards or upwards? Carbon number 1. Beta, think of bird. Correct, upwards. So the OH is pointing upwards. So it's a long, unbranched, very important, long, unbranched, straight beta chain. Now the cellulose, it's got chains. It's got many chains. So I have here, for example, maybe this is a better way. This is one chain. This is another chain. Now this is another chain. So these three chains, they are obviously not physically connected, but they have some sort of bond between them, and we call this hydrogen bond. There are hydrogen bond between these chains, okay, to keep that structure strong. Okay, so we have different chains. When these different chains, when they come together, it forms something called microfibril. So microfibril is made of lots of individual cellulose chain. Right, I'm showing you another image here. So this, I have my, obviously, my cellulose. As you can see here, all of these are beta glucose, beta, and they have straight chain. Okay, so obviously it's going to be glycosidic bond 1,4, not 1,6, 1,4, because there's no branching. And I have chains, cellulose chain. Between these chains are held together by which bond? Hydrogen bond. And that when these chains come together is called microfibril.